So hello again. Good morning, good afternoon, wherever you are. My name is Beverly Baker and I'm Account Coordinator at IDDI. I would like to welcome you to our 10th webinar on the need for better endpoints towards a more efficient drug development in oncology. And we're particularly happy to welcome more than 130 attendees today. So a quick overview of IDDI. Since 1991, IDDI provides biostatistical and e-clinical services in phases 1 to 4 clinical trials. And these include trial design, biomarkers validation, data management, statistical analysis, trial design, medical writing and supporting contacts with the FDA and the EMA. We have developed our own e-clinical suite, which includes an IWRS for patient randomization and treatment allocation, integrated with our EDC system and also internet coding of medical terms. Over the past 20 years, we've developed a thorough experience in oncology, cardiology, ophthalmology, just to name a few. So today's webinar, the contents, with the increasing number of potential targeted therapies, this puts pressure on the ability to evaluate the efficacy of candidate treatments in a fast way. A key element in the evaluation process is the use of sensitive and relevant clinical endpoints. So this webinar will review a range of established and novel endpoints that could be considered for use in cancer trials. We will discuss potential benefits and risks of the use of these endpoints. We will attempt to the extent possible to answer your questions after the webinar. So if you have a question, write it in the chat panel to the right of your screen and send it to us. It would also be great if you could take the time to give us feedback by filling out the survey form, which will pop up after the end of the webinar. Any additional question will be addressed in a follow-up email, and the recording of the webinar will be posted on our website after the presentation. So don't hesitate to contact us for a copy of the presentation if you wish to do so. So who are our speakers today? We have Professor Thomas Bevacizumab, VP of Research at IDDI since 2009. Thomas is Research Professor of Biostatistics, Bioinformatics at the Center of Statistics of Hasselt University since 2002 and serves as Associate Editor for Biometrics. And then we have Dr. Everardo Saad, Founder, Scientific Director and Chair at Dendrix, a medical education, communication and research company based in Sao Paulo, Brazil. And he is also Senior Oncology Consultant at IDDI. So I'll pass you over now to the experts, and I hope you enjoy the webinar.
Good morning, good afternoon. My name is Tomasz, and I would like to present to you the outline of our webinar. We'll start with some definitions, and we will look at current endpoints, discussing their pros and cons. We will then take a look at some proposals for novel endpoints and see how they may be placed in the drug development process and what might be their pros and cons. And then we will look at some tentative conclusions. So I will pass to Everardo to present the first part of the webinar.
Thank you, Tomasz, and hello to all of you attending this webinar. Welcome and thank you for being with us. I'd like to start by trying to define what an endpoint is. Endpoint is one of those words that we all use and understand intuitively, but for which perhaps a clear definition is lacking. Even if we look at regulatory documents, very often the notion of endpoints gets mixed up with the notion of clinical benefit. And although these two things are related, they're not necessarily synonyms. So for the sake of this webinar, I'd like to propose the following definitions. When we refer to endpoints, we're talking about metrics that may represent the efficacy or the safety of variables of interest. In other words, we want to see the effects of drugs, and these effects, of course, may be good. In the case of efficacy or bad in the case of toxicity. For this webinar, we'll concentrate on efficacy endpoints. And most of the time we'll be talking about metastatic cancer because that's where most of the action is in early drug development. When we speak about clinical benefit, we just mean the therapeutic goals that patients, physicians, and regulators, policymakers, and society at large pursue on a daily basis. These two notions are often mixed up, and I think it's important to try and distinguish these two notions. And that's what Dr. Mark Boyce and I have tried to do in this this is what Dr. Mark Boyce and I have tried to do in this recent commentary in the Journal of Clinical Oncology, more specifically in the case of overall survival. This is where most of the confusion is nowadays in my mind, and I guess in many people's minds. So if we consider endpoints as the metrics that we use to assess the activity of drugs, what is the ideal endpoint? Well, as Mark Boyce likes to say, endpoints should capture all clinically relevant events. They should be easy to measure. They should provide little opportunity for ascertainment bias. They should be observed as early and in as many patients as possible. And they should also be statistically sensitive. I'm afraid to say we currently don't have this ideal endpoint. We don't have any endpoint that fulfills all the criteria in the previous slide. So what are the endpoints that we currently have? Well, if we think under the framework where the notions of endpoint and clinical benefit are not immediately untangled from each other, it's very easy to think of a hierarchy according to which overall survival and quality of life sit at the top, whereas the other endpoints stay at the bottom of this pyramid. These other endpoints are often tumor-based, and they include progression-free survival, or PFS, time to tumor progression, or TTP, and the objective response rate. However, over the past few years, it has become clear that a static hierarchy, such as the one I just presented, is not necessarily the best way to go. I like to think of a moving hierarchy in which different endpoints play different roles depending on the disease setting as well as the treatment line. And I hope to make this point clear along my presentation. So let's begin by reviewing the most important endpoints. And I'd like to start with the response rate. Although this is historically a phase 2 trial endpoint, it has often been used in phase 3 trials as a primary endpoint, especially in breast cancer and also in colorectal cancer, not as often in other tumor types. So regardless of the definition used, and of course these days RECIST is used almost always, we say a patient has a response when they have either a complete response or a partial response, and the sum of these two proportions make up the response rate. So the other patients are categorized as non-responders, and that includes stable disease and progressive disease. This has been so for most cytotoxic agents historically. What are the pros and cons of the response rate as an endpoint? Well, it has, I think, an advantage in the sense that it's a very reliable indicator of drug activity. I say that because nothing else will shrink tumors other than active therapy. Whereas if a tumor is growing, you can be sure that treatment is not working most of the times. Also, another advantage is that the sample sizes for trials that use response rate as the primary endpoint are typically smaller than when time to event endpoints are used. However, it has many drawbacks. It's been known for quite a while that measuring responses is fraught with errors because it may lead to errors and bias. Also, for some tumor types and treatment types, lack of response does not necessarily indicate lack of drug activity. And the classical example is hormone therapy for breast cancer. Patients with stable disease also appear to derive some benefit from their treatment. So for these types of therapy, including some targeted therapies, it has been proposed that the disease control or clinical benefit rate may be more appropriate in terms of assessing drug activity. And finally, the association between response rate and overall survival is questionable at best, as we'll see. Let me just give you an example of why response rate may not be so relevant an endpoint with some of the targeted agents. For example, in sorafenib in hepatocellular carcinoma, both in the phase 2 trial and the phase 3 trial on the right, the response rates were very small, in the range of only 2%. However, in the phase 3 trial, there was a comparison with placebo, and there was a 3-month advantage for sorafenib, which clearly indicates that response rate is not capturing the activity of this agent. However, I should like to point out that for some targeted agents, the response rate may be a good indicator of treatment activity. And a very good recent example is that of gefitinib in EGFR mutation positive non-small cell lung cancer. Patients with this type of, with the so-called activating mutations in this gene who are treated with GFR have a 60 to 70% chance of responding. So for that agent in that particular setting, the response rate is able to capture the activity of the drug. Now, there are other ways of using tumor responses to assess treatment activity, and Tomasz will cover a part of that story in his part of the talk. Here, I'd just like to show you in this phase 1 trial of a tyrosine kinase inhibitor. If we look at the top row here, this is colorectal cancer liver metastasis in a patient at baseline before contrast administration, and just a few minutes later there is significant uptake by the liver lesions. And only 2 days after treatment with this tyrosine kinase inhibitor called vatalanib, there is significant decrease in liver uptake, indicating some kind of drug activity on the tumor blood vessels. Also, GIST, or gastrointestinal stromal tumor, is another good example of other ways of assessing responses other than the conventional treatment modalities, which are CT scans and MRIs. So here we have a patient with massive pelvic or abdominal and pelvic lesions that also has very high uptake of glucose, of FDG in the PET scan. So after treatment, one can see that there is pretty rapid and significant decrease in uptake of FDG, which is then followed by measurable response by CT. And in this slide, we can also see another example of the role of PET scanning in GIST. The left-hand panel is the pre- and post-treatment measurement of tumor sizes by CT, whereas the middle panel shows the same thing before and after treatment, but in this case considering the tumor density. And these two measurements are done by CT. If you look at the right-hand panel, it's pretty evident that by using PET scan, it's easier to see a difference between pre- and post therapy situations, indicating that this may be a useful assessment tool for responses in GIST. And in fact, the so-called CHOI criteria are able to discriminate patients with good responses and poor responses as far as their progression-free survival. And this discrimination is pretty much the same as the one obtained by PET scan, and much better than the one obtained by CT scanning alone. So for the so-called CHOI criteria, in addition to tumor volume, the density of the lesion is assessed through the use of Hounsfield units. And this allows for modified criteria which are better able to discriminate between patients with a good and a bad response in terms of their progression-free survival. So talking about this association, let's see the case of surrogacy for overall survival in terms of the ability of response rate to serve as a surrogate for overall survival. There has been extensive work in this regard by Dr. Boyes and Tomasz, who's with me today, and this is one of their published studies. In this study, they looked at individual patient data for 4,000 patients that were included in 27 trials. And these trials compared bolus 5-FU regimens with experimental regimens, and there was an improvement in terms of response rates for the experimental regimens. So they looked at surrogacy at two different levels, at the patient or individual level and at the trial level. And the results are shown here. On the left-hand side, we can see that at the patient level, there is a strong association between responses and survival, such that the odds of surviving is 3 times higher than for responders than for non-responders, considering a landmark of 3 months. Whereas on the right-hand side, we can see that there is a weak association at the trial level, and this is inferred from the finding of an R-squared of only 0.44. And what this is measuring is the association between treatment effects on response rate and treatment effects on survival. So in other words, the variability in treatment effects on response rates explain slightly less than the variability of treatment effects on survival. And this indicates a relatively weak association at the trial level. So talking about overall survival, let's now review this as an endpoint. It's historically the most important endpoint in oncology, and it used to be the most frequent endpoints in most phase 3 trials. However, in recent years, it has become clear that it has some limitations, such that in recent trials, for example, in breast cancer, it's not often used. In fact, it's very rarely used as a primary endpoint, although it's still the most frequently used secondary endpoint in breast cancer. It's still frequent as a primary endpoint in other tumor types, including lung, colorectal, prostate, and gastric cancer, among others. So in terms of the pros of overall survival, it's pretty clear that this is arguably the only objective endpoint in oncology. Perhaps all other endpoints will at some point require judgment or adjudication, whereas there is no doubt about the ascertainment of overall survival. Also, it's the indicator of treatment benefit if we think about the framework in the more static hierarchy that I tried to present earlier. However, overall survival has many cons. One of them is that it requires very prolonged follow-up in comparison with other endpoints, and typically the use of overall survival as the primary endpoint leads to larger sample sizes. And to me, the most important limitation is the third bullet here. Overall survival is influenced by what happens to patients after they progress on trial therapy. And such influence is perhaps the worst limitation of overall survival in those settings for which there is effective second-line therapy. Unfortunately, this is becoming more common in oncology. So there is now this concept of the post-progression survival, which is the time since disease progression and death. And many authors have tried recently to investigate the role or the influence of post-progression survival on overall survival. And among the papers in the literature, this is probably the one that has been cited more often, and in which Broglio and Berry have convincingly argued that the length of post-progression survival, which they called survival post-progression, is important, and as this length gets higher, the chance of finding a significant overall survival gain decreases significantly. And so for many trials that are powered to detect a progression-free survival difference, and a difference is found in terms of median survival, it's possible that such median difference is carried over to overall survival, but the result may no longer be statistically significant just because of the numbers. And this is illustrated in this example. If we think of a situation where there is a gain in PFS of 4 months, and we assume that the same gain carries over to overall survival, which is a relatively optimistic assumption. Only 380 events are required to detect a hazard ratio of 0.75, in other words, a 25% reduction in the risk of progression or death with an 80% power. And using the same assumptions, we would require almost 1,400 deaths to detect a hazard ratio of 0.886. And this shows how less sensitive OS is in comparison with PFS for the same gains in both situations within a trial. However, there are some situations when OS is a good primary endpoint, and a recent example is vemurafenib in melanoma. So in this trial, the drug, the tyrosine kinase inhibitor, was compared with, the RAF inhibitor was compared with this chemotherapy agent, and there was a clear improvement in progression-free survival. Now, since patients until the first interim analysis were not allowed to cross over to the other agent, there was also a significant gain in survival. It's possible that with longer follow-up this gain will diminish just because there will be crossover. But this is a situation where from the lack of post-trial therapy, which are active, a gain in PFS translates into a significant gain in OS. But this is becoming less and less common in oncology. And here I have an example which is exactly the opposite. Sorry, I missed one slide here. This is exactly the opposite example. As many of you know, gefitinib is able to decrease the risks of progression or death in patients with adenocarcinoma of the lung. And this is statistically significant. However, in this same trial with more prolonged follow-up, there was no gain in overall survival, probably because many patients in the control group, those who were treated with chemotherapy in the first line, eventually got a tyrosine kinase inhibitor in second or third line, thus blunting the effect of the drug on overall survival. And this situation is becoming more and more common in oncology. So let's now talk about PFS, which has with time become the chief primary endpoint in some disease types, especially breast cancer. It's also often used as the primary endpoint in colorectal cancer, and there is a trend for increased use in lung cancer and renal cell carcinoma as well. Progression-free survival is defined as the time between randomization and progression or death. I should point out that TTP, or time to tumor progression, has a slightly different definition in the sense that deaths with no prior documentation of disease progression are censored. However, these two endpoints, PFS and TTP, have been often used interchangeably in the literature, so I'm going to refer to them collectively as PFS. PFS has advantages and disadvantages, like all other endpoints, but essentially, they are kind of a mirror image of the case for overall survival. So the advantages of PFS are that it requires a shorter follow-up in comparison with OS. The sample sizes, as we have seen already, are typically smaller. And most of all, it's not influenced by what happens to patients after their disease progresses on trial therapy. On the other hand, it has some drawbacks. Its ascertainment is problematic because it's subject to bias, and in fact it's only a proxy for the true progression date, and it's also questionable as an indicator of treatment benefit. So there has been a lot of work in terms of trying to validate PFS as a surrogate for OS, and this is one of the studies by Mark Boyes and Tomasz. In this study, using phase 3 trials on colorectal cancer, which are represented here in blue circles, so-called historical trials, in which bolus 5-FU was compared with other variations of this regimen, they found a very strong association at the trial level, as we have seen. So treatment effects on overall survival are pretty much very well associated with treatment effects on PFS. And they then tried to sort of validate these results by looking at two types of trials, which they call validation trials. The ones represented by squares here are trials of 5-FU plus irinotecan versus 5-FU alone. And for the two trials which they looked at, the predicted association was there. So these squares sit pretty much on top of this prediction line. Whereas for the oxaliplatin trial, which is represented by this diamond, there was an attenuation of the association. And this is probably explained by the fact that for the irinotecan trials, there was no post-trial use of irinotecan in the control group, whereas for the oxaliplatin arm trial, many patients in the control arm eventually got oxaliplatin after trial participation. So this is a further illustration of the problem I'm trying to allude to in terms of the influence of post-trial therapy. They have also looked at other situations in the attempt to validate PFS as a surrogate for OS, and some attempts have been successful in addition to colorectal cancer. There was a successful attempt in AML among patients in complete remission. However, for many other situations, it has not proven easy to validate PFS as a surrogate for OS using this type of methodology. Now, one may hardly discuss the issue of OS versus PFS nowadays without mentioning the story of bevacizumab. And in my mind, this is a story of a clearly active agent as far as response rates and PFS are concerned, which has not been able to improve the overall survival. So these are the pooled results of 3 phase 3 trials in which chemotherapy was compared with chemotherapy plus bevacizumab, which is the anti-angiogenic agent. It's a monoclonal antibody against VEGF. So what is the reason for this clear improvement in progression-free survival and lack of improvement in overall survival? Well, essentially there are two main possibilities. Either these breast cancer trials of bevacizumab are outliers, or it's also possible that bevacizumab decreases the post-progression survival somehow, and this is under investigation now. However, this is not very common, and at least it's not expected when it comes to cytotoxic agents. And I say that because in other settings, although this is still preliminary, there seems to be no influence on post-progression survival when we think about treatments that are able to improve the progression-free survival. And this is illustrated here by this exploratory exercise. So we looked at 4,400 patients included in 14 trials in advanced colorectal cancer, pretty much the same trials I've been talking about in previous slides. And when we pulled the results of all these trials, and the blue curve here is control treatment and the red curve is experimental treatment, there is a significant improvement in progression-free survival. And the average median improvement is 1.3 months. The same difference carries over to overall survival. So the same, approximately the same difference in overall survival. But interestingly, there is practically no difference in terms of post-progression survival. There are some problems with this type of analysis, and it's not clear what the best way is to assess this variable, but this exploratory exercise suggests that at least for some cytotoxics, drugs that improve the progression-free survival do not have influence on the post-progression survival, and thus we expect gains in progression-free survival pretty much to carry over to overall survival in terms of months, but of course with an attenuated hazard ratio. Let me just say a few words about quality of life. Although it's not very easy to classify it as a purely efficacy endpoint because it also captures toxicity in a way. Quality of life is seldom used as a primary endpoint in phase 3 trials, and as far as I know there is only one case of drug approval, which was gemcitabine many years ago for pancreatic cancer. In terms of its advantage, it's a clear indicator of treatment benefit, and there is no question about that. However, there are many drawbacks of using quality of life as the primary endpoint. Its assessment is very laborious. The instruments that are currently available, despite all the attempts at development and validation, may not be sensitive to some of the issues in this relatively complex set of situations. There are also statistical problems when we use quality of life, mostly because of the frequent missing data, and also there are many correlations between the scores and the domains and things that quality of life attempts to measure. And finally, quality of life, I think, is seldom able to differentiate between competing therapies. So it's problematic as a primary endpoint. So in summary, I would like to show you more green smileys here, but in fact what we have today is a situation more or less like this. For the endpoints that I've been discussing, all of them have problems either with the way they're measured, with their potential for bias, their statistical power, and their clinical relevance. So none of the currently available endpoints are perfect. So let me now hand it to Tomasz, who will hopefully shed some light on this issue.
Thank you, Everardo. So, well, we've heard from Everardo about limitations and advantages of various endpoints, and in view of all this information, there's a question whether there's something else what we could do. And indeed, there were proposals for novel endpoints, and I will try to go through some of them now. But I want to be frank up front, is that it's very difficult to give any firm and conclusive recommendations, because most of these proposals are still under evaluation, and it's always and it still is bound to see whether we can improve on what has been used thus far. So I will be looking at several possibilities, like measuring tumor response on a continuous scale repeatedly, perhaps looking at biomarkers or looking at something which has recently been discussed, which is the time to progression ratio. And also I will come back on the point which Everardo made about biomarkers based on novel imaging techniques. So Everardo did mention several limitations of tumor response. Now, usually tumor response is used as a binary indicator of partial and complete response versus no response. Now, obviously from an information point of view, this is a dramatic loss because we essentially reduce the information about the tumor size to a dichotomous classification at a single point. So we lose both the information about the continuous tumor size as well as about the dynamics of the tumor behavior. These two limitations could be actually amended if we use repeated tumor size measurements, and these are often used in clinical trials and collected. However, they are rarely, if ever, utilized in our assessments of treatment activity. And this is an illustration of what we lose when we use tumor response as binary endpoint. This is a plot from a paper which is referenced at the bottom of the slide, and you can see that it shows the percentage change from baseline in tumor size. And as you can see, only these 3 patients would be classified as partial response because their percentage tumor change crosses the magic threshold of 30%. All these patients would be classified as non-responders, but you can see that there are some patients which clearly had a tumor shrinkage there, and there were others for whom tumor actually grew. So behavior of the tumor in this large group of patients was very much different, and yet we put them into one big group, and based on such a dichotomization, we try to say something about the tumor activity or effect, which seems to be really a wasteful approach to the information we've got. Likewise, we can take a look at the dynamics of the tumor size, and these are the plots taken from this paper, where you can see the waterfall plot, which I've already discussed, but you can also see the percentage change, against coarse number. And as you can see, you can have some patients for whom the tumor clearly shrinks and perhaps starts to grow later, but you have also patients for whom tumor very rapidly grows. And this dynamics aspect is completely missed if we use, for instance, tumor response at 9 months, because all what happens here is actually not captured by this binary indicator. So why not using this information as a potential source of drug activity or even efficacy measure? Now, the idea is to use tumor response as a continuous endpoint is not novel, and this is an example of a paper where the authors looked at the design of a phase 2 cancer trial, randomized phase 2 cancer trial, using a continuous tumor measurement. And this was a trial comparing sorafenib and erlotinib in non-small cell lung cancer. And basically what the authors concluded was that actually treating the change in tumor cells as continuous variable rather than categorizing the changes is a feasible way to design a trial, and actually it is possible to do, and potentially can give more information than just binary indicator. Now, I want to tackle this question about the repeated measurements using a different measure. This is an example of a randomized phase 2 trial in prostate cancer, where patients with elevated PSA after prostatectomy and histological documentation of MUC1 antigen expression were randomized against weekly schedule and 3-weekly schedule of the new drug. Now, one thing which is worth to notice is the small sample size, which actually, from the point of view of using binary indicator, will be prohibitive. One might think that it would be prohibitive to find any meaningful results. And in fact, if we look at the results, the protocol-defined outcomes were PSA response rate, which was defined as the normalization of the PSA levels or the drop by more than 50% of baseline level lasting for at least 4 weeks. Then also duration of PSA response and time to PSA progression. The hypothesis in the trial was that the alternative hypothesis was that the PSA response rate would be at least 25%, or that was what the authors wanted to find. However, in fact, no PSA response was observed on either arm, so clearly in this respect, this trial was absolutely not inconclusive. Now, PSA measurements were collected, during the time of the trial for all patients, and actually we have full pattern of PSA measurements. So the question is whether using this full pattern could actually do something better than by using this full pattern, could we do something better than using the PSA response rate? And actually we can. This is the plot for the two regimens of the log PSA values against time. You have individual patient profiles, and the red lines give the average profiles. Now, what you immediately see from the average profiles is that here's the randomization time. So you see that the dynamics, average dynamics of the PSA, log PSA measurements after the randomization, so when the treatment was included, in the weekly schedule group is very much different than the dynamics which you observe in the 3-weekly schedule. So clearly there's a differential activity in these two regimens. Now what you can see is that approximately the individual patterns of the PSA, log PSA measurements are approximately linear, but deviate from this average trend. So in attempting to analyze this data, we might want to take this into account. And in fact, this is what we can do by using this model. Now, of course, this formula looks ugly, but I will explain it step by step. So we try to explain the logarithm of the PSA measurements by this model, which includes treatment indicator, so the weekly or 3-weekly regimen. We also include the time effect, and this time effect is assumed linear in view of the approximate linearity of the profiles which we've seen on the pictures which I just presented. We also include a potential, individual profiles, so potential for individual patients to deviate from this average linear time trend. And finally, we include in the model the period, this pre and post treatment period, which describes the dynamics of the log PSA measurements in the two periods, and relevant interactions of these factors. Now, these are the results, and again, this is a summary of what we can infer from this model, and I will explain now what we see. Well, what happens is that if we compare the pre-baseline and post-baseline measurements for both schedules combined, so then we see that there's a clear difference between the post-baseline and pre-baseline schedule. So it means that the treatment had an effect on PSA, because essentially post-baseline means measurements under treatment. Now, if we look at the comparison of the post-baseline versus pre-baseline period for the 3-weekly schedule and weekly schedule, we see much more pronounced effect on PSA levels of the weekly schedule. And I would like to remind you that we saw that the slope of the average PSA, log PSA change in the weekly schedule was much shallower than the slope in the 3-weekly schedule. We can see that comparison of the two regimen in pre-baseline measurements is non-significant, but this is no wonder because this was randomized trial, so we would expect that. But if we look at the comparison of the weekly versus 3-weekly regimens post-baseline, there's a clear difference. And actually the weekly schedule had a significantly larger effect on PSA levels as compared with the 3-weekly schedule. So contrary to the results which were based on the PSA response at a particular time point, here by using measurements, repeated measurements of the PSA, we can actually say much more, and we can actually see that these two regimens behave differently and have different activity, which is actually missed if we use endpoints, which are very much limited, and especially in the case of the PSA response, it's a drastic loss of information, as I've explained before. So one could say this would be a potential approach also to tumor size measurements, and obviously this needs to be investigated, but this is a possibility which we should entertain. Now, I will switch to something else which has been discussed for some time. This is using patients as their own control. That is the concept of the time to progression ratio. Well, the general scheme is assume that we have we start treating patients with advanced disease, and on first progression, and we change the treatment to another one, and on second progression we introduce another treatment line, and so on. We could measure time to first progression, time to second progression, and so on, and then we could take the ratios, for instance, of the second time to progression versus the first one, third versus the second, and so on. Now, this is the idea of treating the patient as their own control. This is the idea borrowed from the crossover trials, where we know that treating patients as their own control reduces the variability and increases the power of the trials. Now, the idea is that given the natural history of most tumor, the time to progression ratio generally does not exceed 1. So usually the next time to the next progression is shorter than the time to the previous progression. And Dr. von Hoff suggested that if the time to progression ratio, exceeds 1.33, that this should reflect activity of the second line therapy, because, against this natural history, it seems that the time to next progression is actually longer than the time to the previous one. This idea has been looked at, and this is an example of investigating the activity of higher doses of imatinib for progressing GIST. You can see here the proportion of patients with the tumor with the time to progression ratio greater than this threshold when they crossed over from 400 milligrams to 800 milligrams of imatinib. And as you can see, those who had partial response at 400 milligrams, among those there were 67% of patients who actually, with the increased dose of imatinib, had longer time to the next progression. With the stable disease, there were 36% of the patients who actually had the longer time to the second progression, and then in progressive disease, there were only 17% of these patients. So in total, there was like 25% of the patients who did whose time to progression ratio did exceed this threshold. So it suggested that the switch to the higher dose actually did something good to these patients. This is another example which implements this idea. In this trial, patients who progressed were included in the trial, and then they were subject to molecular profiling by immunohistochemistry, FISH, or microarray. And based on the results of the molecular profiling, the targeted therapy was included, and patients were treated by the appropriate targeted therapy, and the time to the progression on the targeted therapy was measured. Now, the results were that among 66 patients for whom this molecular profiling could be used and actually the targeted therapy could be applied, 18 had this ratio of this threshold for the time to progression ratio exceeded, which was statistically significant. So also the comment was that among the 18 patients, none would have received the same drug through physician's choice. So it seems that this idea of molecular profiling, if we use this endpoint of time to progression ratio, it seems that this idea of using molecular profiling for selecting the therapy after progression might be working. As a side remark, however, let's comment that only randomized trial might prove that molecular profiling does better than physician's choice. This trial was not randomized. And well, a little bit more general comment about using the time to progression ratio is that, well, like in crossover trials, the design becomes inefficient if the correlation between the responses for individual patients is low. And if we look at this proposal from this perspective, this is something which Mark Boyce published. This is where we look at the correlation between time to second progression versus time to the first progression for a group of patients in advanced colorectal cancer. We see that there's very little correlation. So essentially, this idea of using patient as internal control may not necessarily bias much, because looking from this perspective, the efficiency of this design, in view of the lack of the correlation, is not that enhanced. Finally, let us look at the possibility which was mentioned by Everardo already, of using biomarkers which are based possibly on the novel imaging techniques. This is an example of the trial of cediranib, where the primary endpoint was proportion of patients alive and progression-free at 6 months. And additionally, in the trial, magnetic resonance imaging was performed together with some plasma and urinary biomarker evaluations at multiple time points. Now, in this trial, using the designed primary endpoint, what they could discover is that the response rate after cediranib was about 26%. However, when they looked at the radiographic partial responses observed using the magnetic resonance, they observed that the response rate was almost double, like around 57% of 30 evaluable patients using three-dimensional measurements, and it was about the same if they used two-dimensional measurements. Okay, so this means that by using these novel imaging techniques, one could actually come with a measure which captures the effect, or well, actually the activity of the drug much more finely, and perhaps then gives a higher chance to select a drug which might be active in a better way, in the sense that this novel imaging gives a possibility to capture the activity of the treatment better, and therefore gives a chance to retain treatments in the development which otherwise might be dropped by using the standard endpoints. So I think I can offer these tentative conclusions. As I said, it's very difficult to give firm recommendations. As you can see, there are various ideas, and they seem to be promising, but we may need to explore them in more profound way. What we can say is tentatively that all currently available endpoints are imperfect metrics to capture drug activity, as was argued by Everardo. Biomarkers might be statistically more sensitive to this aim. Similarly, the currently available endpoints are imperfect metrics to capture drug efficacy, and especially survival is seldom improved in trials, although it has improved over time in most tumor types. Clearly, evidence from a single primary endpoint may be insufficient for a given trial or setting, and in this respect, I mean, the proposals which I presented of using, for instance, repeated tumor measurements might be at least used as a complementary or secondary argumentation for in a particular trial, to sort of like give an idea about the activity of the drug, which might be measured more finely and more accurately. And clearly, different tumors might call for different development strategies, and most likely, as Everardo was mentioning, one of the key points is the number of active drugs that can be used after the failure on trial treatments, because these second, third, and further line therapies might actually blur the effect of your first line therapy if you are developing one. Okay, so that's basically what I wanted to say. I guess that's also everything what Everardo wanted to say. So I would like to thank you for participating in the webinar. And we have 5 minutes, so if there are questions, perhaps we can attempt. No? Okay. We don't have a question, so it seems that the webinar was clear. Thank you very much for attention, and we invite you to the next webinar, which will be offered by IDDI. Bye-bye.